Hello, this is the Biology 6 lecture on enzymes, and we're going to begin with a review of some concepts and terms that we've already learned about enzymes. We covered enzymes in the last lecture, the lecture on the biological molecules, so we'll start off reviewing a little bit about what we learned about enzymes. So to begin with, enzymes are a type of protein, and proteins are polymers of amino acids. So when you picture an enzyme, picture a big long chain of amino acids. Okay, and we also learned that enzymes perform chemical reactions inside the body. And a chemical reaction is when molecules are changed. And so basically the way these enzymes work is they bind to, each enzyme binds to a certain molecule inside the cell, and then the enzyme performs some sort of chemical reaction on that molecule, and then the enzyme lets that molecule go. All right, so that's the basic concept. Now let's remember a few more things about enzymes. Each enzyme, remember, is a specialist in doing one and only one chemical reaction, or saying the same thing a slightly different way. Each enzyme is specific for binding to only one type of molecule and then doing only one type of chemical reaction on that molecule. So now let's introduce some terms related to all of that. The part of the enzyme where it binds the molecule that it's going to do the chemical reaction on is called the active site of the enzyme. So this spot here, this kind of crevice in the enzyme here, is the active site of the enzyme. The molecule that goes into the active site, in other words, the molecule that the enzyme does its chemical reaction on, is called the substrate of the enzyme. And notice this. Notice that the shape of the active site exactly fits the shape of the substrate. And that's what allows each enzyme to be highly specific. That's what allows each enzyme to bind to only one substrate molecule and do only one type of chemical reaction on that substrate, is the lock and key specificity of exactly how the shape of the active site exactly matches the shape of the substrate. Okay, so when the enzyme has bound the substrate molecule in the enzyme's active site, the next thing that happens is the enzyme does its chemical reaction on the molecule. And in this example, the chemical reaction is to split that substrate molecule into two smaller product molecules. After the enzyme has done the chemical reaction on the molecule, the enzyme releases the product molecule, or molecules in this case. We call those the products of the enzyme. And after the enzyme has released the product molecules, the enzyme's ready to do that all over again. So a new substrate molecule will drift into the enzyme's active site, and the enzyme will do that same chemical reaction on that substrate molecule and release the products, and then another one of those substrate molecules will drift in, and the enzyme again does its reaction and releases the products. So you get the idea. Enzymes just keep doing the same chemical reaction on the same substrate over and over and over and over again. And we call this the enzyme cycle. The enzyme cycle is where the enzyme binds its substrate, does the chemical reaction, releases the product, and then just repeats that. That is the enzyme cycle. Now, from what I've told you so far, you might think that all enzymes' active sites are always exactly shaped to fit their substrate molecules. In other words, you might think that for all enzymes, their active site is always able to perfectly bind the enzyme substrate molecule. And that's usually true, but there are some exceptions to it. With some enzymes, their active site by itself is not able to bind the substrate molecule. And so I'll give you an example. Here's this enzyme here, and here's its active site, and here's the substrate for this enzyme. But as you can see, the active site shape is not perfectly correct to bind the substrate, so by itself this enzyme could not bind its substrate and therefore could not do its chemical reaction on the substrate. Okay, so for some of these enzymes, the ones that their active site is not able by itself not able to bind the substrate, these enzymes have some essentially non-protein helper molecules that are in the active site of the enzyme to help the enzyme bind its substrate molecule. And so there are two types of these non-protein helper molecules that help some enzymes bind their substrate. And the two types are called cofactors and coenzymes. So let's start off talking about cofactors. So this is supposed to be a cofactor right there. There you go. So again, what is a cofactor? Well, it's a non-protein molecule, so it's not made out of amino acids the way the enzyme itself is. It's a non-protein molecule that some enzymes need to keep in their active site to allow them to bind the substrate molecule. And you can see that here. Now that this enzyme has its cofactor, it is able to bind its substrate, and therefore it can perform its chemical reaction on its substrate. So you might be wondering, well, if these cofactors are not proteins, what are they? Well, the answer is metal ions. So that's the definition of a cofactor. It is a metal ion that some enzymes need to keep in their active site so that they can bind their substrate molecule. Here in your web handout for this lecture, I listed some examples of cofactors. Iron ions, magnesium ions, zinc ions. Now, interestingly, what biologists call cofactors, these metal ions that some enzymes need, nutritionists call minerals. Like if you ever take a nutrition class, the instructor might say, every day for proper nutrition, you're supposed to get some vitamins and minerals in your diet. Well, when nutritionists say minerals, that's what we biologists call cofactors, metal ions for some of your enzymes. In fact, here's my bottle of vitamin and mineral supplement. And if you look at the vitamins and minerals that you get out of it, you see that there's a lot of metals listed. Here we go. Iron is one of the minerals in this vitamin and mineral supplement. What other metals? Magnesium, that's a metal. Zinc is a metal. Copper, magnesium, chromium, those are all metals. And there's another one over here, molybdenum, I think that's a metal. Nickel is a metal. Tin is a metal. My God, it's like the whole vitamin pill is made out of metals, right? Yeah, so what I'm saying is those metals that you see on the ingredients to your vitamin and mineral supplement are cofactors that some of your enzymes need, and nutritionists call those metals the minerals. Okay. So to recap, for some enzymes, they need a cofactor, a metal ion, in their active site to help them bind the substrate molecule. Other enzymes, instead of having a metal ion, have an organic molecule to help them bind their substrate. And remember, organic molecule means a molecule that has carbon atoms. And if that's the type of non-protein helper molecule that the enzyme has, we call that a coenzyme. All right, so here's an example of one coenzyme. Not all coenzymes look exactly like this, but you can see this is an organic molecule. And so that's what a cofactor is. It's an organic molecule that the enzyme keeps in its active site to help it bind its substrate molecule. There you go. All right, and I think I have the definition here. Oh, I guess I don't, but it's in your web handout. Coenzymes are small organic molecules that the enzyme keeps in its active site to help it bind its substrate molecule. And nutritionists refer to those as the vitamins. So when your nutrition instructor says every day you're supposed to get some vitamins in your diet for proper health, what nutritionists call vitamins, we biologists call coenzymes. And so here's the label of my vitamin and mineral supplement again, lists vitamin A. That's a coenzyme for some of my enzymes. And vitamin C, that's also a coenzyme. All the vitamins are coenzymes. Okay, well, just to give you an example of one example of a protein in your body that needs cofactors and coenzymes to bind the molecule it binds to. A good example of such a protein is hemoglobin protein. And hemoglobin is a very important protein. It's found inside your red blood cells. So hemoglobin is one of the proteins in your blood. And it has the very important job of binding oxygen. Your blood needs to be able to bind to oxygen so oxygen can be carried throughout your body. And that's the job of hemoglobin, is to bind oxygen molecules. And you can see these things that they showed. It looks kind of a red dot with a white thing around it here and here and here, and there's another one there. There's four of those. Each of those is an iron ion cofactor and a coenzyme called a heme coenzyme. So hemoglobin protein needs a cofactor, is iron ion, and a coenzyme called heme to properly bind oxygen molecules, like you see there. Oh, so incidentally, that's why you need proper iron in your diet for your blood to carry enough oxygen, because this hemoglobin protein in your blood needs that iron ion to help it bind to the oxygens. Or saying the same thing in the reverse way, if your diet is too low in iron, your blood is not going to be able to carry enough oxygen because your hemoglobins can't bind to it properly, and so you're going to get weak from low levels of oxygen in your blood, and that disorder is called anemia. Okay, well, just to summarize, not all enzymes, but some enzymes need either a cofactor or a coenzyme in their active site to help them bind their substrate molecule. Okay, the next topic that we are going to discuss is something called enzyme activity, which can also be called enzyme rate. And it basically means how many times per second the enzyme is able to bind to its substrate and perform its chemical reaction. So kind of think of it as the speed of the enzyme. Okay, so yeah, to understand these concepts of enzyme activity and enzyme rate, let's just imagine one particular enzyme, sitting inside the cell, and those yellowish molecules that are drifting by are supposed to be the substrate molecules for the enzyme. And why are they moving? Well, all molecules just naturally move. All molecules are always in motion. And in fact, you might remember from a previous lecture, I said that the speed that molecules move is affected by the temperature. The higher the temperature, the faster the molecules move. Anyway, so here's this enzyme inside the cell, and those are its substrate molecules drifting around. And so notice that the enzyme is not like a shark where it's going to move around and try to hunt down its substrate molecules. The enzyme itself moves very little. The substrates just have to basically just sort of randomly bump into the enzyme for the enzyme to be able to bind them. But, eventually that's going to happen. Eventually a substrate molecule will happen to drift into the enzyme and fit into its active site. And yeah, then the enzyme will be able to perform its chemical reaction and make the product molecules from the substrate. But again, the point I'm making here is that the molecules just have to sort of randomly run into the enzyme for the enzyme to be able to bind to them and do the chemical reaction on them. Okay, so we define, as it says in your lecture handout for this lecture, we define enzyme activity, also called enzyme rate, as the speed at which the enzyme performs its chemical reaction, meaning how many times per second the enzyme is able to do its chemical reaction. And even though in this cartoon you're seeing here, we don't see the substrate binding to the enzyme at a very high frequency, in real life it happens faster than this. A typical enzyme might be able to bind to its substrate and do its chemical reaction hundreds of times per second, or in some cases even thousands of times per second. But here's the thing. There are various things that can change an enzyme's activity. Various things that might alter how many times per second the enzyme can do its chemical reaction. In the lecture handout, I listed some of these. The temperature is one thing that can change an enzyme's activity. The pH is another thing that can change an enzyme's activity. Changes in the enzyme's amino acid sequence, which we can call mutations, can also change its activity. There are things called competitive inhibitors and allosteric inhibitors and allosteric activators that can all change an enzyme's activity. So what we're going to do is we're going to go through all those factors that I just mentioned, like temperature and pH and the other ones that I mentioned that can change an enzyme's activity, and we're going to talk about those in more detail. But before we launch into the details of how those things can change enzyme activity, I want to mention kind of a general rule. The enzymes in your body don't necessarily have the same conditions as one another in terms of what gives them their highest activity. In other words, not all the enzymes in your body have the same pH that gives them the highest level of activity. But in general, for each enzyme, it's going to have its highest activity, its fastest rate, when the enzyme is in its natural environmental conditions. And what do I mean by that? Well, the different organs of your body can have different conditions inside them. Like, one organ's pH, for example, might be different from another organ's pH. And so what I'm saying is the enzymes where you naturally find them in one specific organ will have their highest level of activity in the environmental conditions inside that organ. And the enzymes in a different organ will have their highest activity, their highest rate, in the environmental conditions that are found inside that organ, the organ where they naturally occur. And so, yeah, I tried to summarize that concept in the web handout by saying in general, each enzyme's highest activity occurs when the enzyme is in its natural environmental conditions, meaning things like the temperature and the pH of the organ where that enzyme is normally found. So I'm saying, as an example, the enzymes in the liver will generally have their highest activity when they're in the environmental conditions that you find inside the liver. And the enzymes in muscles generally have their highest level of activity when this enzyme is in the environmental conditions that you find inside muscles. And the enzymes in your brain generally have their highest level of activity when those enzymes are in the environmental conditions inside your brain. Okay, well, so bearing that in mind, again, that enzymes generally perform best in their normal, natural environmental conditions. Let's now talk about some of these things that can affect an enzyme's activity. And the first one on our list is going to be temperature. So, pretty much your entire body, throughout your entire body, pretty much the temperature is 37 degrees. That's your body temperature. 37 degrees Celsius is your body temperature. So, given that your entire body is 37 degrees Celsius, if I were to ask you, what do you think the temperature optimum for the enzymes of your body is, then you would hopefully say, well, 37 degrees, because you would say, Mr. Edens, you just told us that enzymes generally have their highest activity at their natural environmental conditions. And so for all the enzymes in your body, their natural temperature environmental condition is 37 degrees. And that's pretty much what you find if you studied any enzyme in the body from anywhere in the body, you would find that it has its highest level of activity right around 37 degrees Celsius at body temperature. And here's a chart of that. On this chart, they compared the activity of the enzymes in the body versus what temperature they put the enzyme at. And you can see that for pretty much all these enzymes of the body, right around 37 degrees Celsius, right around body temperature is where they find the enzyme has its highest level of activity. And so if you were to take an enzyme from your body and cool it below 37 degrees, like this part of the graph, the enzyme is less active. The enzyme, is not doing its chemical reaction as much. And likewise, if you were to take an enzyme from your body and heat it up to somewhere above body temperature, like on this part of the graph, then likewise you'd get a lower rate for that enzyme. So again, enzymes, their optimum temperature is body temperature, because that's their environmental condition in regards to temperature. Okay, so what I want to do now is explain this, exactly why is it that enzymes have their highest activity at body temperature and lower activities if you go below body temperature, and also lower activity if you go above body temperature. Why is that? Well, to understand that concept, in other words, to understand the shape of this graph, we need to talk a little bit more about temperature and molecular motion. So I'm going to give an example with water molecules and their motion. So imagine that I take a pot of water right out of fill it up with the kitchen sink, so that's kind of cold to start with, and then I put it on the stove. Well, initially it's cold water, right? As soon as I put it on the stove, it hasn't heated up yet, so it's still cold water. And imagine you could somehow see the water molecules, the cold water molecules inside the pot. That's what I'm going to show them right here. So those little yellow and blue things are supposed to be the water molecules, And all molecules are always in motion, and so those water molecules are bouncing off each other and bouncing off the sides of the pot and also bouncing off the surface of the water. They're in motion. But the point I'm making here is because that water is cold, has a fairly low temperature, the molecules are not moving that quickly because that's what temperature is. It really is a measure of how fast the molecules are moving. But as the pot of water starts to heat up, like this, if you could see the water molecules, they would be moving faster. And so this again illustrates that the higher the temperature, the faster molecules move. And if the water gets even hotter, boy, those molecules are really going to be moving. Again, the concept is that the higher the temperature, the faster molecules move. And incidentally, this is why water boils. Eventually, the speed of the water molecules gets so high that they're able to break through the surface of the water and go up into the air, and that's the steam that you see shooting out of the water when the water is boiling. Okay, so anyway, what you're seeing here applies not only to water molecules, for all molecules. The higher the temperature, the faster the molecules move. Okay, so let's go back to this scenario. So this is supposed to be some enzyme inside the cells of your body, and those are its substrate molecules in yellow that you see drifting around. Now, so the body is 37 degrees, which is a reasonably warm temperature, and so those substrate molecules are moving around at a reasonably fast pace. And so, thanks to the fact that those substrates are moving around fairly quickly, fairly frequently, one of them is just going to happen to run into the enzyme, and so you'll get the reaction happening fairly frequently, like you see there. And so what I'm saying is that at body temperature, the molecules are moving around enough so that fast enough so that the enzyme is able to carry out its reaction at a pretty high activity, at a pretty fast rate. Okay, so now we're going to see, well, what happens if we somehow cool down this enzyme's environment. You'll bring it below 37 degrees. So now we're at a cold temperature. Well, as you know, molecules move more slowly at cold temperatures, right? And so that's going to slow down the activity of this enzyme, right? The enzyme itself is not damaged by the cold, but the substrate molecules that it needs to come to it are going to be moving more slowly. All molecules move more slowly at cold temperatures. So it's just going to take longer for substrates to be able to drift into this enzyme and therefore have the chemical reaction occur on them. And so we're going to get fewer chemical reactions from this enzyme per second, right? And so the activity of this enzyme is going to be lower. So yeah, that's the basis of why lower temperatures slow down the activity of the enzyme. It's just that the substrate molecules are moving more slowly, and so the enzyme is able to do fewer reactions per second. And so on our graph right here, that's this part of the graph, right? At 37 degrees, the molecules are moving quickly enough so the enzyme could do its reaction at a pretty good rate, but the colder you make the enzyme's environment, the more the substrate molecules slow down, and so the lower the activity of the enzyme is going to be. So now let's explore the other half of this graph. Why is it that when we start going above body temperature, we also get a decrease in the enzyme's activity? So at hot temperatures, those substrate molecules are moving very quickly. So at first you might say, well, that should give the enzyme a fast rate, right? Because the molecules are moving so quickly, lots of them are going to be able to go to the enzyme, and the enzyme will do its reaction. But there's a trick. The hotter the temperature, the more likely it is that the enzyme will denature. And denature means unfold. And so what I'm saying is, at high temperatures, there's enough heat energy that it can make an enzyme unfold from its normal shape. And we call that, when that happens to an enzyme, we say that the enzyme has denatured. And what's the effect of that? Well, the enzyme no longer has its proper active site, right? Because only when the enzyme is folded correctly did that active site form in the right shape to bind the substrate molecule. And so, yeah, when an enzyme is denatured, it can't bind its substrate anymore. Its active site just doesn't have the right shape. And so that is why if you start heating an enzyme up above body temperature, the activity of the enzyme goes down. The enzyme gets more likely to be denatured, and when it's denatured, it can't bind its substrate molecules. And incidentally, when an enzyme becomes denatured, the enzyme is permanently denatured. In other words, if we were to take this enzyme and lower it back down to normal body temperature, the enzyme would not refold itself. Again, when enzymes get denatured, they get permanently denatured. Okay, so on that enzyme activity versus temperature graph that we saw a moment ago, just to have the major points, for the enzymes in your body, their optimum activity occurs at body temperature, 37 degrees. And that makes sense because that's the natural environmental condition for temperature, and enzymes generally work best in their natural environmental conditions. If you were to raise your body's enzymes significantly above body temperature, the enzyme has a lower rate because the higher the temperature, the more likely it is that the enzyme will denature, will unfold. If you cool the enzyme below normal body temperature, you also get a lower rate, but not because the enzyme is denatured. The rate is lower just because the molecules are moving more slowly at colder temperatures. Okay, now we are going to talk about the effect of pH on enzyme activity. What we just talked about here was the effect of temperature on enzyme activity. Now let's talk about the effect of pH on enzyme activity. Okay, so let's start off again considering an enzyme, a certain enzyme in one of your cells, and again those yellow molecules drifting around are the substrate of the enzyme, and the enzyme is able to do its reaction on the substrate whenever one of those substrate molecules just happens to drift into the active side of the enzyme, like you see going on here. Okay, but let's say that this enzyme is under its normal natural conditions, and so the rate that you see the enzyme doing here is the enzyme's fastest rate. Well, one of those environmental conditions is the pH of the enzyme's environment. And we talked about pH earlier in the semester. The pH is basically a way of talking about the hydrogen ion concentration of a liquid. And so let's say that this enzyme is at its normal natural pH. It's at the pH of the organ where this enzyme is normally found, and I'm going to represent that normal natural pH for this enzyme with this number of hydrogen ions around the enzyme. So good, yeah. So this enzyme is under its normal environmental conditions, its natural environmental conditions, including pH, and so the enzyme is doing its reaction at a high rate, at its fastest rate. Okay, so now let's imagine we're going to change the pH of this enzyme's environment to see how that affects the enzyme's activity. And we're going to put it in a lower pH than the enzyme's normal pH. And so good review question here. When we say that we're going to put this enzyme at a lower pH than its normal pH, will the enzyme have more or fewer hydrogen ions around it at a lower pH? And the answer is more hydrogen ions, because remember the pH scale is backwards, and that means the lower the pH, the more hydrogen ions. So if we want to now put this enzyme at a pH that's lower than its normal pH, that means we're putting the enzyme in an environment that has a higher number of hydrogen ions than the enzyme normally has. Oh yeah, so what's this going to do to the enzyme? And the answer is it's going to denature the enzyme. Remember the term denature, it means where an enzyme gets unfolded, and a denatured enzyme is not able to bind to its substrate molecules because its active site is no longer the right shape. And when an enzyme gets denatured, it gets permanently denatured, and so this enzyme will never be able to unfold it, never be able to get back to its normal fold. Okay, so what did we learn so far? That to be properly folded, to keep its proper folding, an enzyme has to be at its normal pH. And if you try to put the enzyme at a lower than normal pH, the enzyme will denature, and so that's going to make the enzyme's rate go down, right? And yes, so when enzymes get denatured, they get permanently denatured, but you know that. So now let's start the experiment afresh. I guess we'd have to start with a different copy of that enzyme because that one was denatured. So let's imagine that a new copy of that enzyme, we'll start it at its normal pH. So again, that's the enzyme's natural environmental condition. So the enzyme has a pretty good rate. So now in this experiment, we're going to put the enzyme at a higher pH than its normal pH. And so what does that mean in terms of the hydrogen ions in the enzyme's environment? That means fewer hydrogen ions, right? Because the higher the pH number, the fewer the hydrogen ions. So how's that going to affect the enzyme's rate? And just like we saw before, the rate is going to get less because a higher than normal pH will also denature an enzyme. So you could think of it this way. Enzymes need to be in their normal natural pH environment to stay properly folded. If you move an enzyme to a lower than normal pH or a higher than normal pH, the enzyme will get denatured, and that's going to reduce the rate of that enzyme. So the next slide shows a graph of enzyme activity versus rate. And just let's start off looking at this red part of the graph right here. This red is the activity of an enzyme called salivary amylase, which is found in the salivary glands, the glands in your mouth that make the saliva. And so just focus on that enzyme. So notice that enzyme has its optimum pH, its highest level activity, right around pH 7, right? And so if we shifted that salivary amylase enzyme to a pH that's less than its optimum pH, like around here, what is that, pH 5, it has the enzyme shows less activity because the enzyme is more likely to be denatured. It gets more and more denatured if you go lower than its optimum pH. And likewise, if you take that same enzyme and you put it at a higher pH than its optimum, again, you're going to get a lower activity because, again, enzymes get denatured if you either put them above their normal pH or below their normal pH. Okay, but here's the thing. Different organs in the body have different pHs. And so not all enzymes in your body are going to have the exact same pH optimum as all the other enzymes in your body. So here's a different enzyme in your body. It's called pepsin, and notice that its pH optimum is pH 2. That's where it has its greatest activity. And here's another enzyme in your body. It's called trypsin, and its pH optimum looks around pH 9 or so, right? So even though pepsin enzyme and trypsin enzyme have different pH optimums than salivary amylase enzyme, the idea of what I told you before still holds true. For pepsin enzyme, if you put it at a lower pH than its optimum pH or a higher pH than its optimum pH, it has lower activity because you're denaturing the enzyme. And also for trypsin, same thing. If you put it at a pH that's higher than its optimum pH or lower than its optimum pH, you're going to get a lower level of activity for the enzyme because you're denaturing it. Oh, now here's a thought question. If I were to show you this and say which enzyme of the three enzymes listed here, which enzyme do you think is a stomach enzyme? Would you be able to answer that question? And the answer is pepsin, and here's how you would figure that out. Because the stomach is an acidic organ, there's lots of acids inside your stomach, and acids naturally have low pHs. And so we expect stomach enzymes to have their highest level of activity at acidic pHs like pH 2, right? Because that's the natural environment of the stomach. So we expect the enzymes of the stomach to have their highest activity in that natural environment of pH 2. Trypsin enzyme, notice it has its highest level of activity at pH 9. What organ is that in? The small intestine. The small intestine tends to have a high pH around pH 9, and so same rule. All enzymes show their highest activity at their natural environmental conditions. And for trypsin, which is an enzyme in the small intestine, its pH optimum is pH 9, the pH of the small intestine. Now, one last thing. Let's go back to this graph of enzyme activity versus temperature. And so remember that for enzyme activity versus temperature, you only get the decrease in activity due to denaturing on the higher temperature side. But if you put an enzyme below its optimum temperature, you're not denaturing the enzyme, you're just slowing down the molecules. That's why it has a lower activity on the lower temperature side. Well, we've now talked about how pH affects enzyme activity and also how temperature affects enzyme activity. The next thing we're going to talk about that can affect enzyme activity are mutations in the gene that encodes the enzyme. So here's an enzyme protein here, and you might remember from an earlier lecture that the function of the DNA is to be the recipes for each of your proteins. And we use the term a gene. The gene is one region of your DNA, and each gene is basically a recipe for making one of your proteins. So think of each gene as encoding one protein. And so let's say that this region right here on the DNA is the gene that encodes this particular protein, this particular enzyme right here. Now you may wonder, what are all these G's and C's and A's and T's up here in the DNA? Well, remember that DNA is one of the macromolecules, and all macromolecules are made out of smaller molecules called polymers linked together. Well, the G's and A's and T's and C's are simply the sorry, I think I said that wrong. All macromolecules are polymers, and they are made by linking together smaller molecules called monomers. And so what I was trying to say is the G's and A's and T's and C's you see right here are the monomers of DNA macromolecules. Okay, so if the gene has the right sequence of these G's and A's and T's and C's, then the gene has the right recipe for making the normal functional protein, the normal enzyme here. When we say there's a mutation in a gene, we mean that one of the nucleotide monomers of the gene has been changed. And so keep an eye on this A nucleotide right there. It changed, right? And now it's a different nucleotide. And so we say that is a mutation in the gene. And remember that the gene is the recipe for making the protein. So just like a recipe in your kitchen, if you make a change in it, you're going to get a different food produced, right? And so if there's a change, if there's a mutation in a gene, the amino acids in the protein are going to be changed. And as it turns out, the sequence, the order of the amino acids in a protein is important for the protein to fold properly. So if there's a mutation in the gene, that can cause a change in the amino acids of the protein. And if there's a change in the amino acid sequence of the protein can denature. And so that enzyme is going to obviously have a lower activity right now. Matter of fact, it's not going to have any activity at all because it's denatured. And so yes, if a person has a mutation in one of their genes, they're not going to have a functioning copy of that particular enzyme that's encoded by that gene, because essentially the gene is going to make a denatured version of the enzyme. Well, if a person has that sort of thing where they have a mutation in one of their genes, and so the protein that comes from that gene, they don't have a functioning copy of it because the gene is making a denatured copy, we say that the person has a genetic disease. Genetic disease is where a person inherits a mutated gene from one of their parents, and therefore that person, for one of their proteins, one of their enzymes, is not going to be functioning. Now, so what are the effects of having a genetic disease? Well, that really depends on which gene has been mutated, and therefore which protein the person is missing, because some proteins are more important to the proper functioning of your body than others. So with some genetic diseases, the person is missing a functional copy of one of their proteins, and for some genetic diseases, it's not life-threatening. It might be, kind of an annoyance to go through life with some genetic diseases, but it's not something that's going to kill you. And as an example of one of these non-lethal genetic diseases, color blindness. That's caused by a mutation in a gene that encodes a certain protein that allows some cells in the eye to detect colors properly. Yeah, so people who have color blindness, I'm sure, that's a hard way to go through life, but generally color blindness doesn't kill you. But there are other proteins that are definitely vital to your survival. And so if a person inherits mutated copies of those genes, then the person is not going to be able to survive, at least not to their normal lifespan, because they're missing some vital protein, some vital enzyme inside their body. And let me give you some examples of these lethal genetic diseases. There's a genetic disease called cystic fibrosis. The normal protein, the functional protein, keeps our airways and our digestive organs from clogging up with mucus. And so if a child is born with the cystic fibrosis genetic disease, they have a mutation in that gene, and therefore that protein is not functioning, so it's not keeping their airways and digestive organs free from mucus. And eventually the airways and the mucus builds up to such big levels inside the person that they're just going to die. They can't breathe correctly. And what this woman is doing is she's kind of thumping her child's chest to help dislodge the mucus from the kid's airways. But that's not a cure. Cystic fibrosis is still a fatal disease. So how old do people live if they have cystic fibrosis? Well, if they have top of the line medical treatments, they can sometimes make it into their 40s, but if they're not getting top of the line medical treatments, they can die a lot younger than that. Here's another example of a fatal genetic disease. There's a genetic disease called Tay-Sachs, and the normal functioning protein helps keep nerve cells alive and functioning. But if a baby is born with Tay-Sachs, they've got a mutated version of that gene. They don't have this right protein for keeping the nerve cells functioning properly. And just within a few years, there's enough damage to the nerve cells that the child dies. Babies born with Tay-Sachs disease usually don't make it much past their second year of life. Here's another example. There's a genetic disease called Huntington's disease, and it's a mutation in a protein that's involved for proper functioning of the brain's control of muscles. So when people inherit this mutated version of the disease, they're not able to control their muscles correctly, and it can be fatal also, or it is fatal also. But the people with Huntington's disease usually don't die until their adult years. And let me give one final example of one of these fatal genetic diseases. There's one called muscular dystrophy. The protein that is mutated is a protein that's important for the proper functioning of muscle cells. And so when children are born with this muscular dystrophy, they have a mutated copy of that gene, and so their muscle cells start to die off. And even with top-of-the-line medical treatment, the people born with muscular dystrophy generally don't live much past their 30s. So just to summarize, a enzyme's activity, one of the things that can affect it is whether the gene for that enzyme has a mutation or not. And if the gene does have a mutation, that can lower the enzyme's activity, or in fact just stop the enzyme from functioning at all by essentially denaturing the enzyme. And then the person has a genetic disease, which in some cases can be fatal genetic diseases. Okay, the next thing that we're going to talk about that can change an enzyme's activity are things called competitive inhibitors. Now, to understand what competitive inhibitors are, let's imagine again that this is some enzyme inside a cell, and again those yellow molecules drifting around are the substrate of the enzyme. And remember that the enzyme has to essentially wait till one of these randomly drifting around substrate molecules just happens to have the right trajectory to enter the enzyme's active site, and then the enzyme will do the reaction and turn the substrate into a product molecule. Okay, so to understand the effect of competitive inhibitors on enzyme activity, let me just show you one of these competitive inhibitors. Where is it? Here it is. And so a competitive inhibitor is not the normal substrate of an enzyme, but a competitive inhibitor molecule is close enough in shape to the normal substrate so that the competitive inhibitor can enter the enzyme's active site. But when the competitive inhibitor gets in there, the competitive inhibitor gets stuck. And so what's that going to do to the rate of the enzyme? It's going to decrease the enzyme's activity, right? Oh yeah, so that's the competitive inhibitor, and you can see why. Because the competitive inhibitor gets stuck in there, the normal substrates of the enzyme are just not going to be able to get into the enzyme's active site and do the chemical reaction. The competitive inhibitor is, like you see here, is just blocking the enzyme's active site. So competitive inhibitors decrease the enzyme's activity by basically jamming up the enzyme's active site. And to do that, the competitive inhibitors have to be similar in shape to the enzyme's natural substrate molecule. Now, let me relate this to something that we talked about in a previous lecture. When we talked about receptor proteins, we talked about things called antagonists of the receptor. And an antagonist molecule was a molecule that was not the normal natural molecule that the receptor binds to, but the antagonist for the receptor was a molecule similar in shape to the receptor's normal molecule. And the antagonist molecule got stuck in the binding site of the receptor, and therefore it lowers the receptor's ability to detect molecules for the cell. And so what I'm saying is these competitive inhibitors are the same concept. If you're talking about a molecule that jams up the active site of an enzyme, it gets stuck in there, so it stops the enzyme from doing its normal activity, we call that a competitive inhibitor of the enzyme. But if it's a molecule that jams up the binding site of a receptor, we say that that molecule is an antagonist of the receptor. So I'm just saying receptor antagonists are essentially analogous to enzyme competitive inhibitors. Okay, so competitive inhibitors reduce the activity of an enzyme. There is another thing that can reduce the activity of an enzyme. It's called an allosteric inhibitor. And to understand allosteric inhibitors, let's kind of reset our enzyme here. So again, here's the enzyme inside the cell, and it's just sort of sitting there as these substrate molecules drift by. And when a substrate happens to enter its active site, the enzyme does its chemical reaction on the substrate molecule. Okay, good. So what are these allosteric inhibitors? Well, these allosteric inhibitors, first of all, they bind to the enzyme, but allosteric inhibitors do not bind in the active site of the enzyme. Allosteric inhibitors bind at some other site on the enzyme, not the enzyme's active site. So let's imagine that some allosteric inhibitor comes along, like this molecule right there. So yeah, notice that the allosteric inhibitor, they do not bind to the enzyme's active site because the allosteric inhibitor is not the right shape to fit into the enzyme's active site. So at first you might say, well, that's not going to change the enzyme's activity, right? Because it's not doing anything to the active site. But there's a trick. When an allosteric inhibitor binds to the enzyme, it causes the enzyme to shift its shape in a way that closes the enzyme's active site, like you see here. And so yes, even though allosteric inhibitors don't bind to the enzyme's active site, nevertheless they end up shutting the active site. And so now the enzyme's activity is going to be less, right? Because the enzyme's active site is shut. And incidentally, the word allosteric means different shape. And so that's why they call these allosteric inhibitors, because they are a different shape than the enzyme's substrate, but nevertheless they end up closing the active site of the enzyme. All right, well, there's another thing that can change enzymes' activity called allosteric activators. Let's get rid of that allosteric inhibitor. Allosteric activators are essentially the opposites of allosteric inhibitors. Allosteric activators can bind to the enzyme, but not in the enzyme's active site. But allosteric activators activate the enzyme by opening up its binding site if the binding site is closed. So let's see an example of that. This molecule is an allosteric activator, so it binds not in the enzyme's active site, but when the allosteric activator binds to the enzyme, it changes the enzyme shape in a way that opens up the enzyme's active site, which now is going to increase the enzyme's activity compared to a moment ago when the enzyme's active site was closed. Yeah, so allosteric activators, they increase the activity of an enzyme by opening up the enzyme's active site, but the allosteric activator does not bind to the active site. Okay, let's do a quick summary. So, one of the major focuses of this chapter has been enzyme activity, which is the same as enzyme rate, and that means how fast the enzyme is doing its reaction, how many times per second the enzyme is able to do its reaction. And we talked about various things that can change an enzyme's activity. The temperature and pH of the enzyme's environment can change the enzyme's activity. A mutation in the gene that encodes the enzyme can also change the enzyme's activity. And we've talked about competitive inhibitors and allosteric inhibitors, which can reduce the activity, lower the activity of an enzyme. And we just talked about allosteric activators, which increase the activity of an enzyme. Okay, well, the last topic that we're going to cover in this chapter on enzymes, in this lecture on enzymes, are things called metabolic pathways. To introduce the idea of what a metabolic pathway is, let me start with an example. So imagine we're looking at some molecules that are inside a cell, and one of the molecules we're going to look at is a molecule called glutamate. Here it is, and another molecule that we're going to look at inside the cell is one called proline, a molecule called proline over here. And don't worry about memorizing these structures. I promise you I'm not going to ask you to sketch out glutamate or proline on a quiz or anything. Incidentally, these are amino acids. Remember that amino acids are the building blocks of proteins, the monomers of proteins. Anyway, so let's say these are two molecules that are inside a cell. And let's say, just for the sake of this example, that the cell wants to change some of its glutamate molecule into proline molecule. And why? Well, maybe the cell has a whole bunch of extra glutamate that it's not using, and maybe the cell's having a shortage of proline, and so if the cell could change that glutamate into proline, that's what would be best for the cell. Okay, so you might think, given that the cell wants to do a chemical reaction, to change the glutamate into proline, you might think that, okay, so there's going to be an enzyme, which I'll show as this arrow. You might think, so there's going to be an enzyme that binds glutamate and changes it into proline. And the answer to that is no, and here's why. Each enzyme can only do one small change in a protein. Any given enzyme can only maybe add a few atoms. Sorry, I said that wrong. Each enzyme can only make a small change in its substrate molecule. Each enzyme can only add a few atoms or remove a few atoms from a substrate. But this is a big change, to go from glutamate to proline. And so that's too radical a change. That's too big a change for any one enzyme to do. So to make a big change in a molecule, like you see here, cells have teams of enzymes where each enzyme makes a small change in the molecule and then hands it off to the next enzyme, which makes another small change, which then hands it off to the next enzyme, which does another small change, etc., etc. And so then these enzymes working together as a team can end up making a large change in a molecule. And maybe what I just described to you sounds familiar. It's the same concept behind an assembly line in a factory. In an assembly line in a factory, each worker on the line just does a small change in the thing that's being constructed, and then the object goes off to the next person on the assembly line who makes their own little small change in it, and they hand it to the next person on the assembly line who makes their own small change, and it just works its way down. But with each worker on the assembly line making just a small change, you can end up with a big change in whatever the thing is that's being manufactured. Yeah, so think of when I say the cell has teams of enzymes that work together to make a big change in a molecule. Think of that as like the workers on an assembly line. Okay, well, in that sort of thing inside a cell where you have a team of enzymes working together to make a big change in a molecule is called a metabolic pathway. And for the example that I showed you a moment ago, to change glutamate amino acid into proline amino acid, the metabolic pathway actually has 3 enzymes, which you see illustrated here. And so let's just watch this in order. So the first enzyme binds the glutamate. Here it is. It fits in the active site of enzyme number 1, and then enzyme number 1 makes a small change and then hands the molecule to enzyme number 2, goes into that enzyme's active site. Enzyme number 2 makes a small change, passes off the molecule to enzyme number 3, which makes its own small change, and then what comes out of enzyme number 3 is the proline. So yeah, so this metabolic pathway, these three enzymes working as a team, were able to make that change to change the glutamate into the proline molecule. Okay, well, so that's the essence of what a metabolic pathway is. It's a series of enzymes that bring about large changes in a substrate molecule by each individual enzyme making a small change. Now, in this metabolic pathway that I'm showing you, I actually drew out the enzymes. In most textbooks, when they show you a metabolic pathway, they don't sketch out the enzymes. They just show arrows. Oh, so it's a metabolic pathway. You know that. Anyway, most textbooks, they use arrows to represent the enzymes. So when we look at metabolic pathways for the rest of the semester and you see arrows in the diagram, just realize that those are enzymes. And there are some terms for the molecules that go through the metabolic pathway. So here's the first molecule, that goes into the first enzyme. The molecule that enters a metabolic pathway, the first molecule, that goes into the first enzyme, is called the substrate of the metabolic pathway. And that's the same term that we use for the molecule that enters an enzyme's active site, is the substrate of the enzyme. But now we're saying the first molecule that enters the entire metabolic pathway is the substrate of the metabolic pathway. The molecules that are within the pathway, like this molecule here, are called intermediates of the metabolic pathway. So this molecule is an intermediate of the metabolic pathway. And likewise, this molecule is an intermediate of the metabolic pathway. The final molecule, the molecule that comes out of the metabolic pathway, is called the end product of the metabolic pathway. Okay, so this metabolic pathway that I'm using as an example has three enzymes, these three arrows, 1, 2, 3, and it has one substrate, two intermediates, and one end product molecule. Okay, let me show you a different metabolic pathway. So this metabolic pathway that I'm using as an example has three enzymes in it, right? 1, 2, 3. Most metabolic pathways have more than three enzymes in them. So just as another example, let me show you a metabolic pathway called the glycolysis metabolic pathway. And for today, don't worry about memorizing any of these metabolic pathways specific details. Like you don't have to memorize the names of the substrates or the intermediates or the end products of this metabolic pathway or the one I'm about to show you. I mean, I want you to know what the meaning of the term is, substrate and intermediate and end product for a metabolic pathway, but you don't have to memorize the names or structures of the particular molecules. I'm just using those as examples. But anyway, so here's another metabolic pathway, and again, you don't have to memorize this at all, but it's called the glycolysis metabolic pathway. And the substrate, the molecule that enters this glycolysis metabolic pathway, is glucose. Remember that's our blood sugar. And the first enzyme, you can see this green arrow right here, the first enzyme is called hexokinase. And so the hexokinase enzyme grabs glucose and converts it into a molecule called glucose 6-phosphate. So that's the first intermediate of this glycolysis pathway. Then the next enzyme is called phosphoglucoisomerase, and it changes the glucose 6-phosphate into the next intermediate, which is called fructose 6-phosphate. And then the next enzyme, this next green arrow, is called phosphofructokinase. It changes fructose 6-phosphate into fructose 1,6-diphosphate, the next intermediate of the pathway. Then the next enzyme is called aldolase, and it actually splits the fructose 1,6-diphosphate into two smaller molecules. This one over here is called glyceraldehyde 3-phosphate, and this one is called dihydroxyacetone phosphate. And in fact, this is not even the end of the glycolysis pathway. There are 5 more enzymes that I'm just, for brevity, I'm not going to show you. So the point I'm making here is that metabolic pathways often have several enzymes in them. The glycolysis metabolic pathway has 10 enzymes. Okay, so hopefully you have a a good idea of what a metabolic pathway is. A series of enzymes that working together will make large changes in a substrate molecule. Okay, and cells actually have hundreds of metabolic pathways going on inside them. Oh, sorry, I got a little ahead of myself. Some metabolic pathways are circular metabolic pathways, and we call those a cycle. So this is an example of a cycle. It's called the Krebs cycle, sometimes also called the citric acid cycle. And so the red arrows are the enzymes of this circular metabolic pathway, and they're showing you all the intermediates there. But the point I'm making is if it's a circular metabolic pathway, where the enzymes essentially move the intermediates just around in circles, so to speak, we call that a cycle. A cycle is a circular metabolic pathway. Well, what I was about to say a moment ago, before I interrupted myself, was that cells have hundreds of metabolic pathways going on inside themselves simultaneously. And so this just shows some of these. And again, don't feel you have to memorize any of this, but here's a molecule called, I don't know, what is that? 5-amido-imidazole carboxylate ribulose phosphate. Here's the next enzyme in the metabolic pathway, changing it into N-succinyl carboxamide. I have no idea. Ribophosphate, I have no idea what that is. Then here's the next enzyme in the metabolic pathway that changes that intermediate into some molecule called AICAR. I have no idea what those are. These are some other metabolic pathways. You can see the enzyme arrows down there involving different molecules. And this is just a small close-up of some of the metabolic pathways in cells. Let's zoom out a little bit. And I don't know how well you can see it on your video screen, but yeah, cells have hundreds of metabolic pathways going on simultaneously, and they're crisscrossing with each other. They're feeding intermediates to each other. So, I mean, cells are very complicated in terms of their metabolism, all the reactions that are going on. I mean, my God, this is like a map of the Los Angeles freeway system, right? And so just appreciate that cells have many metabolic pathways going simultaneously on inside themselves all the time. Okay, the next topic is related to metabolic pathways. The next topic is called end product inhibition, or sometimes it's called feedback inhibition. So to introduce this concept of feedback inhibition, of end product inhibition, let's go back to that metabolic pathway that I was showing you before. So this is the metabolic pathway so that the cell can convert this glutamate amino acid into proline amino acid. And you remember how it works. The glutamate binds to the first enzyme that does a small change, and then it passes that to the second enzyme, which does another small change, and that enzyme passes the molecule to the third enzyme, which does another small change, and that ends up making the end product of the pathway, which is called proline amino acid. Okay, so let's say the cell is doing this metabolic pathway, and so it's changing glutamates into prolines. But let's say, for whatever reason, the cell is not using those prolines very much. So those prolines would start to stack up, right? Because the metabolic pathway is running, making a bunch of prolines, but the cell isn't using them. So the cell would want to shut down this metabolic pathway, right? It's just wasteful of the cell's energy and resources if the metabolic pathway is making some end product molecule that the cell is not really using. And so, yeah, if a metabolic pathway's end product is starting to stack up, because the cell's not using it, the cell wants to shut down the metabolic pathway. And cells have a very clever way of shutting down a metabolic pathway if too much of the end product is being made. And that's what end product inhibition is. That's what feedback inhibition is. It's when the metabolic pathway is stopped if the metabolic pathway is making too much of the end product. And so how does the cell do feedback inhibition, if too much of the end product is being made? Well, it's actually quite simple. Feedback inhibition, it works like this. The end product molecule is an allosteric inhibitor of the first enzyme. And so if the end product molecule is not being used, so it's increasing in concentration, it's stacking up, eventually one of those end product molecules will happen to drift over to the first enzyme. And because the end product molecule is an allosteric inhibitor of that first enzyme, the end product will cause the first enzyme to shut its active site, and therefore the first enzyme is not going to be able to bind to the substrate of the metabolic pathway, and that's going to shut down the whole metabolic pathway, right? Because if the first enzyme is not functioning, then there's no intermediate to pass to the second enzyme, and therefore there's nothing to pass to the third enzyme, and therefore there's nothing to be made from the third enzyme. Yeah, so all the cell has to do to shut down the entire pathway is to shut off the first enzyme, and by having the end product of the pathway be an allosteric inhibitor of the first enzyme, that's what happens. The whole pathway shuts down if too much of the end product is being made. Good, and so that whole process that I just described is called feedback inhibition or end product inhibition. And the beauty of it is when the cell starts using that end product, the pathway will start up again. And here's how. Let's imagine that the cell, for whatever reason, suddenly needs to use that proline, that end product. So the cell would start using those end product molecules. So they start vanishing. And eventually the cell would even use the one that's allosterically inhibiting the first enzyme, like you see here. And so as soon as the cell uses that one that's allosterically inhibiting the first enzyme, what happens? Well, that enzyme is no longer allosterically inhibited, so its active site opens up again, and now the pathway starts running again, which is just what the cell wants if the cell starts needing the end product of the pathway. So anyway, I'm very impressed. I've always been very impressed with feedback inhibition. It's just by having the end product be an allosteric inhibitor of the first enzyme, the pathway will essentially automatically shut down if too much of the end product is being made, and the pathway will automatically restart if the cell starts needing that end product. The final topic for this lecture is actually just a review of some concepts that we talked about in a previous lecture. And so the final topic is how enzymes require enzymes that need energy to do their chemical reaction get that energy from ATP molecules. So yeah, this should be a review for everybody. So cells have inside them a high energy molecule called ATP. It stands for adenosine triphosphate. Remember, you don't have to memorize its entire structure, but I do want you to know that each ATP molecule has 3 phosphate ions. One there, and one there, all linked together. I sometimes call that the tail of the ATP molecule. Yes, and so some enzymes need energy to perform their chemical reaction for the cell. And if an enzyme does need energy to perform its chemical reaction, it gets that energy from the ATP molecule. And you might remember, I think I told you this also, this last phosphate of the ATP3 phosphate acts like a cork to keep the energy bottled up inside the ATP molecule. So as long as that last phosphate is still attached to the rest of the molecule, the energy is in the ATP molecule. But if an enzyme breaks off that last phosphate, that's like uncorking a bottle that lets the pressure energy out of it. That releases the energy of the ATP molecule when that last phosphate is removed. Okay, so let's have kind of a zoomed out view of ATP here. And so yeah, ATP is a high energy molecule, and I sometimes try to convey that by drawing a fully charged battery that's just full of energy. Now, ATP doesn't have electrical energy the way a battery does. I'm just making an analogy that ATP is full of energy like a charged up battery is. Good. And yeah, so when an enzyme needs energy to do its chemical reaction, the enzyme grabs hold of an ATP molecule, and to get the energy out of the ATP molecule, the enzyme breaks off that last phosphate, and so you end up with this over here. But that releases the energy, breaking off that last phosphate does. So the enzyme gets the energy that it needs to do its chemical reaction, and what's left over is called adenosine diphosphate. Diphosphate means 2 phosphates, and an unattached phosphate ion that's not part of any organic molecule anymore, and that's sometimes written P subscript I. It stands for phosphate inorganic because it's not connected to any organic molecule anymore. Anyway, so when an enzyme uses ATP for energy, what's the enzyme gets the energy, but what's left over is ADP and a PI, an inorganic phosphate, just a phosphate group by itself. And those are low energy because, remember, after that phosphate has been removed, the energy has been uncorked, so to speak, and so these are now low energy molecules. Okay, so the enzymes in the cell are constantly using the cell's ATP to get the energy that the enzymes need to function. So if there was no way to recharge ATPs, in other words, if there was no way to reattach that phosphate and get it back to ATP, then the cell would just run out of ATP eventually, right? But that wouldn't be good because the enzymes would just stop from lack of energy. So the cell does have a way of recharging its ATP supply. The cell does have a way of reattaching that last phosphate to change this back into ATP. And I tried to show that as kind of a green arrow here. But that takes energy, right? If you have a battery that's been drained of energy, it takes energy to recharge it to its high energy form. And so it takes energy to reattach that last phosphate back on and make the ADP back into ATP. Where does that energy come from? Glucose is the answer. Cells take in glucose from the blood, and the cells use the energy in the glucose sugar to recharge their ATP supply. And so, oh, sorry, I didn't want to go there yet. Go back. There, yeah, cells use glucose, the energy in glucose sugar, to recharge their ATP supply. Now, here in this diagram, I've been emphasizing the ATP is the energy source for enzyme proteins, but other proteins other than enzymes sometimes also require energy. So other proteins other than enzymes can also use ATP for energy. And just to give an example of that, this is supposed to be a channel protein. Remember that channel proteins are proteins in the cell membrane that can move solutes across the cell membrane. And so let's say that the cell wants to move one of these solutes inside, let's say this solute, to the outside. So that solute, I guess it's this solute molecule, would go and bind to the binding site in the channel protein. But let's say that this channel protein requires energy to move that solute across the cell membrane. Well, just like enzymes do, channel proteins that need energy can use ATP as an energy supply. So that channel protein would then bind ATP, and by changing that ATP into ADP, getting the energy out of it, the channel protein would get the energy it needs to pump that solute out of the cell. Okay, but getting back to this diagram, so ATP is the high energy molecule that supplies energy to enzymes and also to other proteins in the cell that need energy, and the cell uses the energy in glucose to recharge its ATP supply. Okay, well, that is the end of this lecture on enzymes. And so, as usual, I advise you to do the review questions on this lecture topic, and I will see you in the next lecture.